So hey guys, welcome to this session. Myself, Dr. Varsha Gupta, right, on the behalf of eGurukul, I am here in front of you and we will be discussing the recall question that for the exam that happened yesterday, INI-CET May 2022. So up on your screen is my Telegram ID. If you have any thing like any more information, if you want to correct my questions also, please feel free to send me the message. Also, you can pause the video and put the comments up there that there was these options that were there. Okay, so based upon my analysis and based upon my interaction with the students, I could figure it out that biochemistry portion was relatively very easy, very straightforward questions. For those who have done very good, like the genetics in the +2 in the high school, they have found it like comparatively easier, right? And I even like, it was amazing that how straightforward this question exam like paper could be. It is more of towards the genetics portion, right, rather than taking everything in account, right? But my suggestion for you too is for the upcoming NEET PG exam, don't think that you have to just remember this way. Don't change your strategies so easily just based upon INI-CET. Okay, just continue with the studies. Remember this, okay, whatever you are doing, right, the entire holistically for the entire all the subjects, you are gonna pass the exam, not just one subject. Okay, so definitely notes given to you by all the faculties, all the renowned faculties are gonna make you pass the exam. You have to just focus on your revision. Okay, so there was this one question about the maple syrup urine disorder, right? So I was not sure, students were not sure either. They were saying the not restricted or restricted, which one is restricted. So what we should know about the basic concept is, as we always talk about, that maple syrup urine disorders is isoleucine, leucine, valine, right? I love Varsha ma'am, right? The branch chain amino acids. Amino acids are the issues those say should not be given to the diet. So only option left is methionine. Sometimes you don't have to think much. The question answer might not be the one you have ever even heard on, but you can eliminate it, because the not just based upon your knowledge you can pass the exam. You need to know how to take the exam. So these, we call it as exam taking techniques. Try to eliminate the incorrect options. That will give you a better chance to pick the right option, right? So next question, there was this question that uracil was found in the genetic code, right? And we know that methylated version of the uracil is the thymine, right? It should be there in the DNA version is the thymine, but uracil was found. So what could be which vitamin deficiency is leading to? Very straightforward, like definitely most of you have picked the right. But if I don't know the question, I don't know the answer, how we are going to figure it out? We know that there is a difference of just the methyl group between the uracil and the thymine, right? If methyl is not there, it means who is the methyl donor? Do we know the methyl donor? Only two methyl donors we talk about, the SAM and the THF, right? THF is tetrahydrofolate. If you don't know even the fully activated form of THF, that is N5N10 methylene tetrahydrofolate, you know this is FF is folate, and folate kind of similar words is there. So you can even guess the answer. Okay, now there was this question that bending technique is used in dicentric chromosome, right? Very straightforward. C is for the chromosome, right? Match the following transporters with the molecule they carry, right? This is one of the another question. So there was these choices we have to match. So heme is carried by hemopexin, heptoglobulin is carried like carries the hemoglobin, pre-albumin carries the thyroxine, and albumin carries the free fatty acids. Albumin also carries vitamins, hormones as well. Okay, so this is the choice, that one is A, two is B. Like the way it was given to me was already a correct sequence, right? So you can match your answer. Now there was this question, a question on genetic engineering steps. Again, the students were not sure, for those whom I have interacted, right? So the main theme of the question was to order, align, like make the arrangement right. Step 1, step 2, step 3, step 4. How do we do it, right? The vaguely they were giving me the options like there was an option of lysis of bacteria, gene of interest, protein extraction, expression, SDS page, like so many options were there and they were vague, right? So we know what are the major steps of genetic engineering, right? Those are, first of all, we have to identify the section, right, which we have to work upon. Then we have to extract that gene, right, and insert that particular gene into a vector, right, and then we multiply it. Why we multiply it? By inserting the plasmid into the host cell. So host cell is a natural environment, and then we can make the so many copies of that gene of interest. So whatever was there in the options, which I don't know, if you know it, please tell me so that I can explain you better. You can figure the sequencing out, right? And the options were vague. And there is one pretty much very interesting thing I could have figured it out since like last 2-3 years when I'm interacting with the students. What I'm getting is that there is not single version of a question paper. In question paper, they are giving, let's say this in a genetic engineering steps, they have multiple forms of the same questions. So that one student is getting one form and another student is getting second form, right? That is why there are so many options which come to me and I am unable to figure it out. And the students are pretty sure, ma'am, they have seen this, right? So probably one question which have two different forms, and few students are getting one form of a question, another are getting another set of form of the questions, but the essence or theme of the question is the same. So you should be knowing the holistically everything what is going on, right? So if there was a protein thing, protein extract Kia, like express Kia, SDS page Kia, we know that SDS page is for definitely for protein, right? And to have, why do we use the SDS page? Because this sodium do-decyl sulfate is a detergent, right? So protein can have any kind of a charge, positively, negatively charged. So this sodium do-decyl sulfate has excess of the charge, which masks the protein's charge. So overall, all the proteins, when you are separating the protein, because this is a protein separation technique, so overall charge on the protein is, and all kind of proteins are the same. So when you do the extraction, charge is not the case by which they are being extracting. We extract them based upon the molecular weight first, right? Then we run the molecular amide jet. So if you know the concept behind, you should be able to figure out the answer. Since I don't have the right sequences or right choices for it, I am unable to give it. But if there were the stages of sequences of the genetic engineering, so these are different stages. First, you should know the DNA gene you are interested in. Then you take it out, right? Then you put it into a plasmid. Then you put it into a vector. Vector is like an environment where you want it to multiply. Same, if you can imagine this with the mammals, right? They mate, right? They mate, so you have to put male and female at one place, right? So vector is that place, right? And plasmid is gonna go there and multiply or divide it into various copies. That's how we do the genetic engineering. Okay, now this was the question about the KM-VMAX Michaelis-Menten question. Very, very standard question. It's a fixed question. As I always say, biochemistry is a highly scoring subject. बहुत आसानी से score कर सकते हैं. The reason being, most of the questions are just obvious. You don't have to think it. If you know it, you will get it right. Right. So same is the Michaelis-Menten. We have the graph. We know the graphs of the competitive, non-competitive inhibitor. And I was very glad to see this question because I have just discussed it in recall questions, right? And we know this is the line we were worked for. Probably something X was given, right? X was given to me, right? That what the student said, ma'am, X was given to me. And we know this is the line we were worked version of the Michaelis-Menten, because we get the accuracy of the numbers are much more, right? And this I have discussed it, right, just few days before the exam. So for those who have watched the video, they probably have gotten it right. So the X is, if you say X means no, right? No is a competitive inhibition. And what is the trick? Whenever there is a competition, max, right? Competition, max, always increases. Competition में max always gets increases. That's how you took it. This is 0, right? This is 0, this is negative. So more it is towards the 0 side, the bigger the number is, right? So that is the competitive inhibition. In the upcoming exam, you may not get competitive, but you might get the uncompetitive, right? Or non-competitive. Uncompetition, they are not compatible enough. So they always run parallel. They always run parallel. Uncompetitive runs parallel. And non-competitive. Non-competitive means nobody is in the competition. They are working very well. Both the substrates and inhibitors काम कर रहे हैं अपना, but they are not competing. Like I call it myself as non-competitor. I don't compete with anybody, nor I compare myself with anybody. So remember, Varsha ma'am is a non-competitor. I think my competition is just with me only. I don't compete with anybody else. Even I don't compare myself with anybody else, right? So V is non-competitive, and if the K max increases, right, competition में K max को बढ़ जाता है, that is going to be a competitive inhibition, right? So the choice was competitive, right? Maybe the A option was there. Which of the following is false regarding Human Genome Project? Human Genome Project was the biggest project of all, like till today, the scientists ने जितना सबसे बड़ा project लिया is Human Genome Project, right? The options were like the collaboration of many countries. Yes, many countries collaborated into it, right? And few students were giving me the various kind of numbers. Total genes were identified. I was not sure what that number could be. It is a modern it uses the modern sequencing technique. Definitely itna bada project hai. They used these techniques. It took 2 years, maybe 2 or maybe 12, but 2 years to discover. But what are the important facts? Important facts, one is down there. It started in October 1990, right? And it ended in April 2003. Okay, so this is the exact time date from directly from the website, right? And what was the major, like 3 ways to determine everything, how they bifurcated the entire project, right? First of all, determine the order or the sequence of all the bases, right? Then make the maps that which gene is located where, and then produce the linkage map that this gene is linked to this gene is linked to this gene. That is how they figured the entire human genome out, right? And it took approximately 13 years, right? And it is a publicly funded project in which many countries contributed, and more than 2,800 researchers, they took part in it, and many countries were involved, and many research centers were there in the US, UK, Japan, France, Germany, China, and many other countries, right? An entire human genome is 3.3 billion base pairs. Now there is one more line in which in some places in the Human Genome Project, they claim it to be 3.3 billion, but the Wikipedia is saying 3164.7 million. So mind this, which word they are giving you, whether they are giving you a billion or a million, right? So it is, sometimes they write it 3.3, 3.1, both are correct. And I'm pretty sure they are never gonna give you this 3.1 or 3.2, like both of them. But if they do give you this, I'll suggest you to pick 3.3. Okay, on average, there are approximately 3,000 nucleotides, and proteins are encoded only by the 2% of the genome. Rest extra is the garbage, and that extra garbage, or you can say, since there are so many duplicate genes that are available, that are helping the scientists and researchers to figure out how we are evolving from the primordial times to the current time, right? So most of the genome is a repetitive sequence with no coding purpose. So these are all the facts of the Human Genome Project. There was this question about the deamination of methylated cytosine will form. Again, I got lucky, and my students also got lucky in that, because we have discussed this questions in the recall sessions, right? Methylated version of the cytosine will form what, right? This we have discussed. This is methylated cytosine, that cytosine having the methyl group. Let me quickly change the color of my highlighter. I'll pick the black color now. So this is the cytosine down there, and this is the methylated version of the cytosine, right? If you remove the N from here, so definitely it is going to be a keto group, but there will be methyl group here. If you don't know even the structure, Amino group is gone, right? Cytosine amino group is gone. There will be keto group and methyl is there. Which of the these figures down there have the methyl group? Only the thiamine has a methyl group. Only the thiamine has a methyl group, right? So methylated cytosine is the thiamine. And another way to figure this question out is cytosine is what? We know there are two kind of these nucleotides, purines and the pyrimidines, right? Another way to look at the question. Purine is adenine and the guanine, right? And pyrimidine is cytosine, uracil, and the thiamine. These three are the pyrimides. The question is asking the question is about the cytosine. So definitely purine is not gonna be the option. So A and G, you can cross the out. They have different structure, right? The structure is not changing. We are just removing one functional group out. So uracil and thiamine you are left with, but they are talking about the methylated version. Uracil don't have the methyl group. Thiamine do have a methyl group, right? If you don't even know much about the structure, just based upon few facts, based upon few basic knowledge, you can figure the answer out, okay? The icosanoids are formed from what, right? Again, in the previous recall questions, this is like many times they are asking it. Every year they are asking the question revolving around the arachidonic acid. Sometimes they ask you the enzyme name, sometimes they ask you something else. This time they have asked you the icosanoids are formed from. What are icosanoids? They are made from the 20 carbon, right? Polyunsaturated fatty acids, that is majorly your arachidonic acid, right? They are what? They are the signaling molecules. Your prostaglandins, thromboxane, leukotrienes, lipoxins, all these are your icosanoids, right? So, and they are made from the arachidonic acid, right? So our answer to this will be A, that is arachidonic acid. And rest options I don't have. If you have it, please do share it with me in the comment box or just share it with me in my Telegram group, right? Which of the following is correctly matched, right? So you have the oxygen, glucose, calcium, sodium, right? Oxygen is by simple diffusion, yes. Glucose is transported by facilitated diffusion, sodium dependent, right? Calcium is by active transport, sodium is by secondary active transport. All seems right to me. Maybe like few options are missing, maybe information is missing. But do remember that calcium transport is both by the active transport and also there is a receptor mediated. Hold on, I guess I have somebody who is buzzing me, right? Let me just quickly take a look at it, okay? Well, guys, so I'm back here. Calcium is via the active transport as well as the receptor mediated, that is passive. Even the sodium is both the sodium active transport and there is a passive transport to, or you can say more of it is a channel transport, right? Where do we see it? You see it in the sodium potassium pump. You see it, that is kind of a channel. Then you see the sodium ATPase, right? That is a secondary active transport. So if this is given and they are asking you, maybe for me all are correct. Probably there was a second half portion of this question in which you have to say only A is correct, only B is correct, all are correct. To me all are correct, but know the extra knowledge that calcium is both active as well as passive, that is the active transport, and also the receptor mediated, which is a passive transport. And sodium transport is the secondary active transport and also the via the channel, that is a passive transport, right? Sodium potassium pump and the sodium ATPase pump is there, okay? Best way to identify gene mutation if it is less number of alleles are there, right? So I am not pretty sure whether they are phrasing this question in a right way or not. I'm not very much sure, right? But if we know about the techniques, what do we know about the PCR, right? PCR technique. How do we do that? Polymerase chain reaction is done when you have very small quantity of maybe in the nanogram of the DNA, but you know the sequence, right? You have very small quantity And majorly it is used for what? To test whether a particular foreign kind of a genome, maybe let's say the virus, since it is a COVID era we are living in, is present or not, and how much it is present, the quantity. That is why sometimes it is known as qPCR, quantitative PCR, right? qPCR, it is said as qPCR. And Q is usually written a smaller alphabet, right? So that is telling you whether a particular virus is present or not in general, right? And the double droplet is kind of a PCR, but it is a more advanced version of a PCR. Usually in the PCR, if there is some kind of another element that is present in the sample that acts as an inhibitor, so it is degrading your result, it is masking your result. So in the double droplet PCR, this is more accurate, more better quantification is done, right? So that is what PCR is. Why do we use the DNA fingerprinting? Choice B. DNA fingerprinting usually is related to for the paternity testing, right? If you have to find a culprit, sample DNA is with us, right? We have to find the culprit. So we have to compare the samples of the two people, whether this is this person or this is this person, right? So that is the DNA fingerprinting is used for. Fish is fluorescence in situ hybridization. Fluorescence in situ hybridization, you take a probe, right? You take a probe, and probe is like you imagine it is like in ancient times hota tha na ki there is this beautiful lady and the swamber is going to happen and he's going to look for the bride, is going to look for the groom. So fluorescent probe is beautifully decorated girl, right? And she is going to go and look for the match, right? So who accurately matches it? All the matches, all the samples, whatever are there, right? They are your grooms, and bride will pick only one guy and will get married to it or will stick to it, and then she's gonna fluoresce. Fluorescing is the marriage. Okay. So if they are talking about just one person and we have to find the best match, because allele is what? Allele is a genetic variation of a gene. Gene a single hai, that is the wild type we call it. If there is a little bit sequencing changes there, then we call it as an allele, right? So if we have to find an allele, we should be making exact probe which will not stick with the allele, right? So for me, it will be the fish. But if you have maybe more elaborate explanation or like question ka kuch aur portion bhi hai, just give me that so that I can get a better clue, right, to it. Oncogene marker, again, I have just discussed it in the recall question. Probably they were asking for the neuroblastoma, and I said that neuroblastoma N is M by C. When you have C, right, then what is BC? BC is Burkitt. BC is Burkitt lymphoma, right? Able gene. Able gene is what? AB. AB के बाद क्या आता है? ABC, right? So it is CML. It is CML. RAS is for many cancer, but especially it sticks with the prostate cancer, right? Very simple. DDP, DDNTP, dideoxy, right? There was a similar kind of a question I have taken in the recall versions. This is this question, right, in which we talk about dideoxynucleotide. Okay? In like deoxynucleotide, you have OH at 2', right? In dideoxy, you don't have OH. In the Sanger technique is to know, to determine the nucleotide sequence, right? Ki kaun sa nucleotide sequence is there or not, right? That what Sanger technique is doing. So what we have to do is we have to take something which will interrupt the sequencing, elongation sequencing technique. So jahan interrupt hoega, we will be having all sizes of the fragments, and then we read that fragment. And what do we call the Sanger technique? Sanger technique ko hum kya bolte hain? Sanger technique is also known as chain termination. And who is helping to terminate the chain? ठीक है, कौन काटेगा गला sequence का, right? कौन terminate करेगा? That is your dideoxynucleotide. Dideoxynucleotide that does the termination of the polymerases, does not allow polymerase to work. Okay. And with this, I would like to thank you, and I wish you a very good luck for the upcoming exam. Don't worry, don't get nervous. You all are warriors. You are not just the corona warriors, but you are the warriors of your life. And I'm pretty sure you're going to make it. Know the concept, revise everything. Don't get confused कि INI-CET may they have asked this, so probably upcoming NEET PG they are going to ask the similar kind of the questions. They may or they may not, who knows, right? Who knows, we don't know yet. So stick with the strategy you are following. Don't get confused and just go for it. And remember, if there are changes in the times of the uncertainty, hold your own hand, because this is your war, this is your life, and you have to fight for it. Okay, kal kya ho raha hai, nahi ho raha hai, ye sab ho raha hai, wo sab, so much going on out there in the world. So what if they are changing the examination pattern? Yes, there is a first frustration, but remember, what I have learned about in my life is, like my life experience says, is when you are frustrated about something, that decreases your capability. So never ever get frustrated about anything. Okay, time was good, then few years are going bad, but again good times will come. That right? Not everything is changeable, right? समय का सिद्धांत है बदलना, और वो बदलेगा. Good days are gonna come out, and though that time only your knowledge is going to help you, nothing else is gonna help you. So just keep focusing on your studies and keep yourself happy. Engage with people who make you happy, because if your body is happy, your mind is happy, then you can be able to grasp more from your studies. Okay, with this I would like to take a leave. Please leave me message on my Telegram group. I can share you that again on my Telegram group, or just leave me a message, email me. You can email me, right? Whatever suits you the best. My Telegram ID is @ Dr. Varsha Gupta, right? That is, I'll write it down. So this is my Telegram ID, right? @ Dr. Varsha Gupta. If you can scan it from the screen, you can scan my code also. So I will take a leave. Thank you.